Hi there, my name is Ricky Hill, and today we're going to talk about a fascinating intersection of nutrition and neuroscience, the potential for the ketogenic diet to support recovery from traumatic brain injury, or TBI. TBI is one of the most complex neurological disorders, impacting nearly every domain of function: cognition, mood, behavior, motor control, and autonomic regulation. In moderate to severe cases, injury often results in cellular energy failure, widespread inflammation, and structural loss, particularly in the prefrontal cortex, hippocampus, and white matter tracts. At the cellular level, TBI initiates what's called the metabolic injury cascade. The brain's demand for glucose spikes, while its ability to process that glucose plummets due to mitochondrial dysfunction and oxidative stress. This leaves neurons starved for fuel when they need it most. Even long after the acute phase, patients often experience persistent memory loss, executive dysfunction, mood instability, and in some cases, progressive neurodegeneration. So clearly, we need more than rest and rehab, and this is where the ketogenic diet, or as we'll abbreviate it, KD, enters the conversation, not as a miracle cure, but as a metabolic therapy designed to shift the brain's energy strategy. So what is the ketogenic diet? Why might it help? KD is a high-fat, very low-carb diet that mimics the metabolic state of fasting. Normally, our bodies convert carbohydrates into glucose for energy, but when carbs are restricted, like in ketosis, the liver starts breaking down fats into ketone bodies, primarily beta-hydroxybutyrate, acetoacetate, and acetone. These ketones can cross the blood-brain barrier and become an alternative energy source for neurons, which is especially valuable when glucose metabolism is impaired, as it often is after TBI. What's truly fascinating is how this shift influences multiple systems. Ketones enter mitochondria and generate ATP more efficiently than glucose, while producing fewer reactive oxygen species. Ketosis also enhances mitochondrial biogenesis, promotes GABAergic signaling, which calms hyperexcitable neurons and upregulates antioxidant defenses like glutathione. And on the hormonal level, KD can modulate leptin and insulin, reducing systemic inflammation and potentially improving recovery signaling cascades. And the result? A brain that is more resilient to injury and more capable of repairing itself on the molecular level. So let's dig into the research. We'll start with animal studies, where most of the experimental evidence exists. In a series of rodent models, KD led to significant improvements in motor coordination, spatial memory, and reduced lesion volume after induced TBI. Some other studies found that these benefits were age-dependent, with younger rodents showing more pronounced neuroprotection. One standout mechanism was the preservation of brain volume, including protection of oligodendrocytes, the cells that produce myelin, myelin sheaths, which insulate axons, and axonal mitochondria, which are typically destroyed after injury. Beyond behavioral outcomes, studies identified several neurological targets. Mitochondrial support. KD upregulated PGC1 alpha, or get ready, it's a mouthful, peroxisome proliferator activated receptor gamma coactivator 1 alpha, which is a master regulator of mitochondrial biogenesis. Basically, it helps cells grow new mitochondria. It also increased uncoupling proteins, UCPs, which are protective proteins that help reduce harmful byproducts like reactive oxygen species. Excitotoxicity prevention. KD elevated GABA, the brain's main inhibitory neurotransmitter, while reducing glutamate, the brain's primary excitatory neurotransmitter. This helps balance out overactive neurons and prevents cell damage from overstimulation. Neurogenesis. KD stimulated brain-derived neurotrophic factor, BDNF, and nerve growth factor, NGF, which are proteins that support the growth and survival of neurons. And several studies even pointed to modulation of the gut-brain axis. After TBI, gut dysbiosis, or an imbalance in gut microbes, can worsen behavioral symptoms. KD was found to restore gut integrity by increasing tight junction proteins, which help keep the gut barrier intact. This may reduce inflammation and improve communication between the gut and the brain. And although human studies are limited, we do have emerging evidence. One study demonstrated feasibility of KD in subacute brain injury patients, with markers of improved ATP production and cognitive gains. Other studies highlighted the potential for nutritional therapies to influence recovery trajectories, though they do call for more controlled trials. And a final study in a Department of Defense review concluded that further animal studies and eventual human randomized controlled trials are warranted given the mechanistic process. So how strong is the case for keto? We have a strong preclinical foundation showing neuroprotection across physical, behavioral, and cognitive domains. Mechanistically, KD targets core vulnerabilities of the injured brain: mitochondrial dysfunction, inflammation, and excitotoxicity. There's evidence of synergistic effects. For example, KD combined with BCAAs further reduced seizure activity in animal models. But we also need to be honest about the limitations. Most studies are in animals, not humans. The evidence is limited in diversity. Many rodent studies use only young, healthy males, which doesn't reflect real-world populations. And KD can be hard to follow long-term, with potential side effects like constipation, hyperlipidemia, and adherence fatigue. So should we discount it? Not necessarily. Here's the rebuttal. Animal studies are essential first steps. Most FDA-approved neurological drugs start with rodent models, and as one study points out, rodent genes share approximately 95% similarity with humans. Modified diets or exogenous ketones may provide the same benefits with fewer barriers. These include MCT oil-based approaches or targeted fasting protocols. The safety profile is acceptable in supervised settings. Ketogenic diets have been used successfully in epilepsy for decades, especially in children, where long-term follow-up shows sustainable benefit. And most importantly, no existing therapy directly targets the metabolic dysfunction following TBI. KD might fill that gap. So what does this all mean? We're starting to understand TBI not just as a mechanical injury, but as a metabolic crisis. The ketogenic diet offers a way to re-engineer the brain's energy supply, reduce oxidative damage, and even promote cellular repair. This could be particularly transformative in early recovery when glucose metabolism is impaired, or in younger patients who may respond more robustly to ketone metabolism, and possibly even as a preventative strategy for high-risk groups like military personnel or athletes. But we need more data. Human randomized trials exploring timing, dose, and tolerability. Comparative studies on modified versus classical ketogenic protocols. Long-term studies on neurodegeneration prevention, especially as some TBIs are now understood as progressive. In conclusion, the ketogenic diet represents more than a trendy weight loss strategy. In the context of TBI, it is a serious metabolic intervention with the potential to restore energy balance, mitigate damage, and enhance neuroplasticity. We're not quite ready to make it standard care, but with the evidence that we do have, it deserves serious clinical attention. Because when the brain is running on empty, it may just need a different kind of fuel.